We interrupt this program to bring you a report about antisense therapy and gene therapy, two scientific procedures which are on the rise and may be the next paradigm in treating physical illnesses, or maybe procedures with disadvantages that outweigh their benefits. Let's take a look at how these therapies work to begin. Antisense therapy is a process of gene silencing and protein aggregation. During the process of protein synthesis, the translation step is prevented. What happens is, copies of mRNA are inserted into the cells containing a disease-causing gene. When the mRNA from the disease-causing gene is going to the ribosome for translation, the antisense mRNA binds to it, and translation cannot occur, meaning that no disease-causing protein will be created as a result. The disease-causing gene is considered to be turned off. The antisense mRNA is a mirror image of the mRNA that it is inhibiting, allowing it to bind to that specific mRNA. Gene therapy is a similar process. It is the insertion of normal or genetically altered genes into cells to replace defective or diseased genes. The inserted gene will either compensate for an abnormal gene or make beneficial proteins. The genes cannot be directly inserted into cells, so it is necessary to work through vectors. A vector may consist of a modified virus to carry the gene into the cell. Also, cells from the patient may be taken out, have the vector placed inside them, and then returned to the patient. Vectors can be injected or inserted intravenously. In gene therapy, DNA is directly altered by the new incoming genes, whereas in antisense therapy, the mRNA produced from DNA is altered by the new incoming mRNA. Now, these technologies may seem like a significant advancement, and while they do certainly have their benefits, there are several disadvantages as well. Let's take a deeper look into the advantages and disadvantages of these technologies. A primary aspect where antisense therapy trumps other traditional drugs is its specificity. The antisense mRNA is designed to only interact with its corresponding disease-carrying mRNA. Once this interaction has occurred, all disease proteins that could have been created were prevented. Traditional drugs tend to target every disease protein that has already been created. This antisense treatment only needs to inhibit one mRNA to prevent the many proteins that would be created otherwise. Both therapies tend to be more efficient than antibodies. The two procedures have more permanent effects and get right to the base of the issue, as opposed to the antibodies which only target the diseased proteins that have already been created. Lastly, these treatments could serve as a source of hope for patients with certain diseases. As we will see later, there have been cases where these therapies have succeeded and cured diseases within patients who otherwise may have not had a chance at surviving. The half-life of the drugs involved in the procedures are usually only around 24 hours, meaning that multiple treatments may be required and consequently a lot of money is needed. In antisense therapy, the inserted mRNA needs to be a very specific size so as to penetrate the cell, bind to the mRNA, and avoid nucleases. Otherwise, the treatment will be unsuccessful. As the drugs involved in the procedures are usually nucleotides, both will face the problem of nucleases. To avoid this problem, the drugs are typically genetically modified to last longer, which may allow the treatment to work, but which is unhealthy to the cell. An ethical issue is the topic of playing God, or playing nature. If these treatments are available, humans may never have to evolve and develop natural immunities to certain diseases. It is argued that nature should be allowed to take its course. Being that the treatment would likely be expensive, it may only be available to the rich and upper class. This is another ethical dilemma because not everyone would have access to the therapies. Lastly, the two therapies are still very upcoming, and there are many unknowns surrounding them. They are being researched heavily, with some successes, but it is still unclear. The procedures could even trigger a severe immune response within the body, as the drugs can be targeted as foreign molecules and attacked by the immune system. So, the general reaction towards these upcoming biotechnologies? We asked the opinion of a professional. During my time at Harvard Medical School, Harvard Medical School, I was really able to study antisense and gene therapy. I think the two treatments are a fantastic idea and could certainly become a common practice in the future. We know that back in the 90s, we experienced the first successful treatment of gene therapy. A young 4-year-old girl who suffered from adenosine deaminase deficiency was given a gene therapy treatment consisting of removing her white blood cells and inserting them with the adenosine deaminase. It had successful results, but they were only temporary. This practice has been done on other ADA patients, and there were similar results. While there have been successful occurrences, I still do not believe that this should become a regular practice. In fact, it cannot. It is still being researched and tested on human and animal patients. Until the drugs are considered completely safe and will benefit the patient, the therapies cannot become full-time practices. Aside from that, however, the financial issues need to be resolved, and people could certainly misuse the drugs. We're just not ready for these therapies quite yet. We still need to research these treatments more, obtain successful results, and then resolve the ethical dilemmas. Then, we can look into this becoming a common treatment within many clinics around the country. Well, there we have it, folks. A scientist's perspective on gene and antisense therapy. Now, you may be wondering if the two treatments are legal or not. Here in Canada, there are not many regulations addressing the two treatments. However, it is stated that before any drugs can be put into practice, they need to be tested and produce sufficient evidence that they are safe, efficient, and fully functioning. In the US, the drugs are being researched more heavily, with more tests being conducted. Full regulation there, but it is known that the drugs are not available for sale. There are, however, more opportunities for antisense therapy in the US and Canada. We will conclude our show with kudos to the scientists we believe deserve them for their contributions to the two therapies. Paul Zemechnik is often considered the inventor of antisense therapy. He studied protein chemistry and attended Harvard Medical School. Zemechnik also co-founded tRNA and worked in his lab studying oligonucleotides. Stanfield Roger was a scientist who first proposed that good DNA could be used to replace the defective DNA within human cells. Several scientists tried to attempt Roger's proposal, but it wasn't until the late 80s that Dr. French Anderson performed the first successful experiment on a patient involving gene therapy. As a matter of fact, Anderson performed this treatment on the 4-year-old girl with the ADA disorder. This has been Isaac Heading, reporting about antisense therapy and gene therapy. Good night, everyone.